Microbes exist almost all over environment. They are small, but highly diversified and important for various metabolic activities, immunity for hosts, and many other processes. But unfortunately, many of microbial species are difficult to culture in the laboratory. So, metagenomic sequencing approach was invented. This approach conducts DNA sequencing of whole community genomes to know the member composition and function in environment. This is a brief summary of metagenomic analysis method. I will briefly explain metagenome and related words. Microflora, microbiota, microbial community mean total collection of microorganisms within a community. Metagenome means total genomic potential of a community. Microbiome means microbiota and metagenome in a microbial community. Actually, microbiome is an ambiguous concept. In general, when we talk about metagenomic analysis, we often mean two types of analysis method. One is Amplicon sequencing analysis. In the case of prokaryotes, Amplicon sequencing analysis uses 16S ribosomal RNA gene in most cases. This is a summary of Amplicon sequencing workflow. Extract DNA from a community, PCR amplification of so-called phylogenetic marker gene, sequencing and analyzing the member composition of the community. 16S ribosomal RNA gene is commonly used for Amplicon sequencing because this gene has many good properties. For example, all prokaryotes have this gene. This gene has a mosaic structure of conserved regions and variable regions, among tasks. In general, evolutionary history of 16S ribosomal RNA gene corresponds evolutionary history of the species. So, this gene is very useful for phylogenetic marker genes. Other method is so-called shotgun metagenomic sequencing, or simply called metagenomic sequencing. The experiment procedure is simple. Extract DNA and sequencing without PCR, but the bioinformatics method is complicated. After conducting the complicated bioinformatics analysis workflow, you can get member composition and functional composition of microbes. Here, I will explain the importance of DNA extraction. Using appropriate DNA extraction method is very important because both of Amplicon and metagenomic sequencing analysis use DNA extraction. Since microbes have diverse structure outside the cell membrane, for example, gram-positive bacteria have peptidoglycan layer, and gram-negative bacteria have small peptidoglycan layers and outer membrane layer. So the outside the cell membrane structure is very different. There is no best DNA extraction method that can be used for every microbes. So it is necessary to select an appropriate extraction method depends on the types of microbial community, like gut microbes or soil microbes or water microbes, with referring previous studies. This choice of the DNA extraction method is very, very important because we cannot go back after obtained sequence data. In the end of the 2022, about 4.5 million microbial sequencing data were deposited in the INSDC sequence read archive. So there are already huge amount of public Amplicon sequencing and metagenomic sequencing data you can use. This is the detailed stats of the public microbiome sequencing samples. human microbiome, especially human gut microbiome sequencing sample, constitute large proportions. But other environments, sequencing samples are also existent, not a small amount of, not a small number of samples. For example, soil, already 0.5 million samples are deposited. And most of them are Amplicon sequencing sample. And shotgun metagenic sample is not so large numbers. But in human gut microbiome, already many research papers suggesting the relationships between human genome and gut microbiome and human phenotype, including disease. For example, this table that was modified for me from these papers. The study list of more than 1,000 samples with analyzing human genome and gut microbiome association. There are already many examples exist from a large number of the sample with analyzing both of the human genome. Human genome in this study, slide, the meaning is that human SNP dataset and human gut microbiome composition. Although we still don't know whether the gut microbiomes are causation of some human phenotype like disease, many researchers conducting Amplicon or metagenomic sequencing of human microbiome like this.